All right. So folks, I'd like to take a few minutes before we have a little break and talk a little bit about what I asked you to look up yesterday. Now we might take just sort of as a given that DNA is the molecule of heredity, but that was not always the case. Until not realistically that long ago, we didn't even know that DNA existed. And people had lots of different ideas about what parents were actually passing to their offspring that was making them resemble their parents, so offspring resembling parents. And so the experiments that I asked you to read about or to look up were both experiments that were trying to determine what was the molecule of heredity, what were parents passing to offspring. So, I asked you to summarize first Griffith and Avery, and then Hershey and Chase, really by focusing on what their experiment was. Now, if I could summarize what Griffith and then Avery were doing, these are two different guys. One came first, kind of died, the other one kept on working on the same sort of idea. This is the gist of what they were doing. You can see in my picture, there's some mice, and then on the bottom row, either they're standing upright, which means they lived, or they're flipped over, which means that they did not. And they did a bunch of different tests where, you can see there's a little syringe, they injected different stuff into the mice to see if they lived or died. So, in the first trial, they injected what they called non-virulent strains. So, ones that didn't have the properties of a virus, and the mouse lived. Then, in the next round, they injected the virulent ones, the ones that have the virus-like property, and the mouse is flipped over. Then, they did a couple of other tests where first they heat-treated things. Heat is a well-known way of killing things or denaturing things or making molecules not work the way that they should. Depending on how much you remember Bio30, maybe you remember like proteins don't like heat, and if you heat them up, it changes their structure so that they don't act the way they're supposed to anymore. So they figured if they heat treat stuff and kill things, that should work. And oh, good, mouse survived. Then in the last one, they kind of mixed the heat treating and the non-virulent strain together. What they did essentially was they took, see these blue ones, the non-virulent strain, and they mixed it with the heat-treated, the ones that should have been inactivated. And somehow, the inactivated ones transferred their properties to the non-virulent strain. Now, I'm never going to ask you to repeat the details of this experiment like you have memorized them, but I would, and so would the diploma, describe an experiment like this to you and then ask you, what was their manipulated variable? What was their responding variable? And what might be a controlled variable? So that's what I want to ask you. And you're going to tell us what was their manipulated variable. Correct. What they injected into the mice changed in each trial of the experiment, and they were changing it on purpose to see what would happen. That's the definition of manipulated variable. The one the experimenter changes on purpose to see if it has an effect. Now, what was their responding variable? Yeah, reaction of the mouse, survival of the mouse, whether the mouse lived or died, something to that effect. Now, what might have been a controlled variable? Sometimes you got to infer these. Yeah.
The species used.
Yeah, species, for sure. I don't want to do this on two different species of organism because they might have different levels of resistance to these virulent strains of bacteria that I'm injecting into them. If you said anything about the type of mouse or the initial health of the mouse, I would say that's a good controlled variable. Like, you don't want a mouse that was already going to die in 5 minutes, and you don't know if it died because of your treatment or because it was just old. So if you said like initial age and health or size of the mouse, like all of these things are factors that you want to keep constant. I did a bit of research when I was in university, and we worked with rats in our lab, and it was like a super big deal that we always only used male Sprague Dolly rats of this specific type that we only got from this specific place. They had to be a particular weight, so I had to weigh and feed them every single day, twice a day, weekends, holidays, whatever, and then clean up their poop. It was very glamorous. Anyways, those types of things need to be controlled. Now, if we turn to what conclusion did they draw from their observation, something like this. Something like organisms can pass characteristics on to other organisms. They called it the transforming principle. It's not a vocabulary word that you have to be able to define. That's just what they happened to find in their particular experiment. So the idea of organisms passing stuff on to other organisms via the molecules that they are made of. That's the idea that we should be getting from this. Now, the other experiment that I asked you to read about was Hershey and Chase. Hershey and Chase came a little bit later on, and they were investigating very specifically which molecule gets passed on. The consensus, or what a lot of people thought, was that it was proteins. Proteins got discovered first, and it seemed like proteins were what was getting passed on. But they checked to see, is it protein or is it DNA? So they took these bacteriophages. Those are viruses that infect bacteria. For one of them, they labeled protein with a radioactive marker so that they could follow where did the protein from this virus go. In another trial, they labeled the DNA from that virus green. Well, okay, let's go back. It's green on this picture. But they labeled it with a different radioactive marker so that they could follow where did the DNA go. On the left, you see the results of the trial where the protein was labeled. When they labeled the protein coat of that bacteriophage, they found that when it infected a bacterial cell, the protein didn't go inside of the bacteria. It stayed on the outside. When they labeled the DNA and they let the bacteriophage infect the bacterial cell, they found that the DNA was actually going inside the bacteria. And so they determined that DNA was what was getting passed on. This is a pretty classic experiment here. So let's do the variables. Could somebody tell me what the manipulated variable is? What did they change on purpose to see an effect? The marker, precisely. What they marked, which structure was marked. And what was the responding variable? Yeah. Yeah. The bacterial cell. Yeah, I know it looks like an egg on this picture because it's low quality art, but you're right. Whether or not the marker went inside of the cell or stayed outside of the cell. So you could say the final location of the molecule that they marked or something like that. And then a control variable, well, think of the same thing we just said for the mice. Make sure I'm using the same type of bacterial cell, same strain of bacteriophage, same initial age and health of all of the cells involved, so we can be confident that whatever happened because of what we did, and not because of some random other factor. Now, does anyone want to ask anything about either of those? Yeah. Control variable, we could say, like, the type of bacterial cell, the type of bacteriophage, initial size, age, health of either of the types of cells we're using. Anybody else have questions there? Okay. Let's take a little break here. So the next thing which really I could have put all as the same lesson, is now what does DNA and this mysterious thing called RNA that I'll tell you about, what's their structure? We know from those scientists and their research that people studied things to determine whether or not DNA is getting passed on. And when we realized it was, we figured, well, we've got to figure out how this works if this is the molecule that's getting passed on. Now, I've added to my title here who the scientist was who isolated them. That means separated them from other chemical structures, like in a chemistry lab. Levine didn't figure out how DNA works. He just figured out, hey, this is the molecule that we should be studying. And so I'll mention scientists from time to time and who did what. And once in a while, there'll be a question asking you, hey, which scientist goes with which discovery? You wouldn't have to memorize, like I said, with those scientists we looked at, like what were all the steps that they went through. But anytime I find it worthy to mention a scientist, it probably means that you should at least have a vague idea of the big picture of what they did. Okay? Levine's first name is Phoebus, which I also find entertaining, and so maybe you'll remember him because Phoebus is a fun name to have. So, these little structures are called DNA nucleotides. Now, just as a side note, does anyone know what DNA stands for? With a big, long, tricky thing. The D is deoxy, or deoxyribo. The N is nucleic, and the A is acid. I won't ask you that. Neither will Nicole. We'll call it DNA all the time. But it's called deoxyribonucleic acid because of the structure that it has. Now, each one of these little structures is what we call a nucleotide. And Levine is the one who isolated these little chemical structures. Now, you might say, okay, I thought DNA was this like big, long chain. And it is. Of a whole bunch of these nucleotides attached to each other, then there are two strands, but we're starting with the basic pieces first. There is, on every DNA nucleotide, okay, this one first. Sorry, we'll go back. I guess I can't use green. There is a 5-carbon sugar. Oh, I can't use red either. Okay, never mind. I'll just point. So, if I take a look at the structure, do you see this little pentagon with a D in it? That's the 5-carbon sugar. It is a pentagon, has 5 sides, and the sugar has 5 carbons, so the 5 kind of goes together with that. And it has a D in it because the 5-carbon sugar is deoxyribose. Now, I wanted to write that word, but I guess I'll just have to write it over here. Oh, no, I won't. There it is. Deoxyribose. I wish I could write that on the smartboard, but it's not letting me use the ink. But that's the name of the 5-carbon sugar. That's why it has a D. Now, the second thing that every DNA nucleotide has is a phosphate group. So you'll see there's a little circle with a P in it on each of the nucleotides. Those are the phosphate groups. Now, there's a line between the sugar, the deoxyribose, and the phosphate, and it represents a chemical bond. And this is not a chemistry course, and I'm not going to ask you to name all of these types of bonds, but just keep in mind that there's actually quite a bit of chemistry involved in this. If we have time today, we're going to watch a little clip where they show some of this animated. And a lot of the videos will give you a bit more chemistry details than maybe you need to know. Just know that if you have to know it, I will have said it in the notes. If I didn't say it, it means that it's cool, interesting chemistry, but you wouldn't be tested on it in this course. Now, the last part of the DNA nucleotide is called a nitrogen base. So there's a part at the top. I'm pretending to circle it because every time I touch the smartboard, it advances the slide. But in the first nucleotide, there's an A, and then there's a hexagon. In the second one, there's a T. Third one, there's a C. Fourth one, there's a G. Now, that's 4 nucleotides. But if you look at my description, I'm saying there's actually 5. But only 4 of them are part of DNA. There's a 5th one that's not part of DNA. So I'm focusing on the DNA ones here. The names of these nitrogen bases are adenine, guanine, cytosine, thymine. And if you look at the nitrogen bases, the part at the top, they all have a letter in them. A for adenine, T for thymine, C for cytosine, G for guanine. Now, would you have to draw this from memory on a test? No, you would not. But you should recognize what the components of a nucleotide are. They all have phosphates. They all have that 5-carbon sugar. They all have a nitrogen base. Okay? If you look at the nitrogen bases, they're not all the same size. And that brings me to my next couple of points. At first, when we discovered these nucleotides, people thought that a person's DNA just went ATCG, 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 forever and ever, and that was the sequence. That there was 25% A, 25% T, 25% C, 25% G. But it's not. A guy came along called Chargaff. He's an important guy because he came up with this particular idea. First, he said it's not constant. It doesn't always go A, T, C, G, and then repeat itself. And those 4 nucleotides are not in equal quantities. There's not 25% of each of them in every person's cell's DNA. What he did discover, though, is the last point. Even though they're not all equal to each other, the amount of adenine is always equal to thymine. So A and T are present in equal quantities. And then guanine, cytosine. So that means adenine and thymine go together, guanine and cytosine go together. And if you look at them, like A is a bigger nitrogen base, T is a bit smaller. C is a bit smaller, G is a bit bigger. Oh, that's handy. If I pair them up, A with T and C with G, I get the same sort of size of pair, a big one with a small one. Now, does anyone want to ask anything right there? Okay. I want to show you an example. I really wanted to write on the board. I don't know if I'm going to be able to. Ooh, okay, I think it's going to let me write. Okay. So I'd like to do a little example to show you what you might have to do with Chargaff's rule. Chargaff's rule is about A and T being equal, C and G being equal. So here's the kind of thing that we might ask you. I might say in a particular sample, oh, it's way down there. This smartboard is the worst. That brings us to what DNA looks like once we put those nucleotides together. We call DNA a double helix. Double for 2, helix because it's twisty, DNA, I always say, looks like a twisted ladder. It's got two sides, it's got pieces in between, and it's twisted. So if you imagine like a curved staircase, like the ones that a lot of people with an elevated deck will have going down to their yard, that's what DNA kind of looks like, is a curved staircase or a curved ladder. If I had taught you junior high, when I taught junior high 100 years ago, I used to always make DNA. You could try this at home. I took licorice for the sides and put a piece of string through it, and I went red, black, red, black. That's what the edge of DNA looks like. Then I would take gummy bears on a toothpick and make the rungs with it, and then twist it, and you had an edible piece of DNA double helix. That's just an aside. We are going to build DNA models once we talk about how DNA replication happens at the molecular level. So on Monday, we'll spend some time playing with models where you're actually going to put some DNA together. Now, there's a few important people. Rosalind Franklin, who doesn't get enough credit, she took all sorts of X-ray photos. She collected an enormous amount of data. But then Watson and Crick, these two guys, came along, and they didn't really do experiments themselves necessarily. They collected data that other people had measured and figured out what DNA must be like based on other people's observations. So Watson and Crick get a lot of credit because they made a model of DNA. They're very famous for it. But Rosalind Franklin is actually the one who figured out all of the data that is letting them come to these conclusions. Anyways, lady scientists didn't get a lot of credit back in the day. Hopefully things are different now. But here's some of the things that they determined. First, DNA is a double helix. I just explained what that meant. It has sugar phosphates on the outside. So if I look at my DNA, I've got two different versions of a picture here. Neither of them is super molecularly accurate, because both of them are just art representations trying to show you the pieces. The picture on the top is showing you on the edges sort of an alternating striped pattern. That's to represent sugar phosphate, sugar phosphate, sugar phosphate. So when you look at this picture on the top, and you can see on the outer parts, it looks like it's alternating stripes. That's to represent sugar phosphate. On the picture on the bottom, the green circles are the phosphates, and the orange pentagons are the sugars. And so what we're trying to get at is on the outside, or the backbone, we have sugar, phosphate, sugar, phosphate. The nitrogen bases are always on the inside. So on the picture on the top, we're using different shapes to represent them to kind of give you the idea that the nitrogen bases fit together nicely with only one other nitrogen base. So I said A and T are always present in equal proportions. That's because they're complementary to each other. C and G are always present in the same quantity because they're complementary to each other. So if you look at the bottom picture first, you'll see that when I have A on one side, I have to have T on the other. Whenever I have G on one side, I have to have C on the other. So anytime a nucleotide is present on one side, its complement has to be on the other side. Now I'm using a couple of words here, purine and pyrimidine. And I'll be giving you guys data sheets, probably, maybe I'll bring them tomorrow. You don't really need them super bad right now, but purines are what we call the A and the G. And so if you remember, and I'll just go back to the last slide, so you can see how A and G kind of look the same? T and C are the pyrimidines. They kind of look the same to each other. And what I'm saying on this next slide is that we're making complementary pairs. We're always taking a purine, which is one of the kind of bigger looking ones, and matching it up with a pyrimidine, which is one of the smaller ones. And they are attached by hydrogen bonds. sorry, the pyrimidines are the smaller ones, and the pyrimidines are the C and the T. So purines are the A and the G, pyrimidines are the C and the T. Okay. Now they're attached by hydrogen bonds. I don't suppose anyone remembers what those are from a chem class that you might have taken some time ago. It's a Chem 20, I think. Anyone wants to guess? So lots of, oh yeah, yeah. Take a stab. Yeah. Or were you gonna ask me a question?
No.
Okay, so yes. So it's when hydrogen is attached to a really electronegative element, like nitrogen or oxygen. In this case, nitrogen, because it's a nitrogen base. Essentially, what happens is the really electronegative element steals all the electrons, so hydrogen basically has none, giving it a very strong, attractive, positive force to other things. You don't have to explain what a hydrogen bond is. But some of you probably learned that in Chem 20, right? It's part of Chem 20. And so I just want you to know that's what it is. Essentially, it's not quite as strong as a chemical bond, but it's a really strong attractive force. So we call it an intermolecular force. So it's not a chemical bond where two atoms are sharing electrons, but it's an attractive force nonetheless. So in the top picture, the three little dots, or in the bottom picture, the dashed lines, those represent hydrogen bonds. It's not a full bond, that's not why it's not a straight line, but it's a strong attraction. The last thing here is that the two strands are anti-parallel. This is a little hard to explain without explaining chemistry. So I'm going to try to explain just a smidge of chemistry here so that it makes more sense. We call the two strands anti-parallel because their backbones, so the sides, have opposite directions. And we always use this 5', 3' jazz. If you take bio in university and you're doing genetics, you might get into this a bit more. I just want you to know that it is not the main focus of Biology 30. The 5 and the 3 are referring to the way that you number carbon atoms in organic chemistry. I'm going to bet that most of you have not done Chem 30 yet, right? And so you wouldn't have even learned even a tiny bit about this. So I'm just trying to give you the overview. There's a whole branch of chemistry called organic chemistry that deals with molecules with carbon atoms as their base. Proteins, sugars, acids, there are lots of molecules that are related to biology that are all carbon-based. Okay? We are carbon-based life forms. All the molecules that make us up are carbon-based. Okay? So, when we have molecules that are made out of a bunch of carbons, so that we can correctly name them, we say that each carbon atom has a number, so that we could say, well, it's carbon number 1 or carbon number 2. Okay? So this 5 and 3 with a little apostrophe, the apostrophe means prime, is us counting which carbon atom stuff is attached to. That's where the numbers are coming from. Now, do you have to be able to explain 5 and 3 prime? You do not, but it gets used in a lot of biology descriptions, and I just want you to know, all it's saying is the way the backbone is oriented is like flipped. The bottom picture does a really good job of displaying that. Do you see how on the left, the pentagons are pointing up, and on the right, they're pointing down? That's all we're trying to get at, is the backbone is oriented in the opposite direction. Now, would anyone like to ask a question here? Okay. I'm going to pause my recording because I'm going to show you more.